Yeah. I was like, hi, everyone. Thank you so much for being here. For those folks in the room, thank you so much. Hopefully you're enjoying your lunch. For folks online, hopefully there's someplace comfortable and you're also getting some food. But my name is Cinnamon Moffett. I'm the research program manager here at Hatfield Marine Science Center in Newport, Oregon. And I'm excited that we have the opportunity to host a lunch and learn. So for folks that are familiar with Hatfield, we do a Thursday seminar. Lunch and learns are an opportunity to take advantage of guests that come and visit us that might sit outside of that seminar schedule. So for today, we have a great lunch and learn. I'm going to hand this off to the person who connected us to today's speaker.
Hi, everyone online and in the room. I'm Jessica Garwood, and so I've gotten to meet Elizabeth Brasseale, our speaker today, through the coastal oceanography community. We do very similar work. Today, she will tell you about how wind stress affects the source of ocean inflow to the Salish Sea and the implications for invasive larval connectivity. To know a bit more about Elizabeth, she's done her PhD at UW with Parker McCready before doing a postdoc at Scripps with Sarah Giddings. And now she is back at UW, so she's visiting us from Seattle, and she's working with the eDNA Collaborative in the School of Marine and Environmental Affairs. And she won't discuss that work today, but because she just started that postdoc, but she would be really happy to interact with people in the room or here at Hatfield about that project and talk about it more casually. So with that, Elizabeth, take it away.
Thank you so much. And okay, second to I was told not to move around, so I probably don't need to. Oh, almost down. It's not going to be okay.
That might be
better. Okay. Hi, everybody. I'm I'm Elizabeth. Thanks very much for the introduction, Jess, and for arranging everything, Cinnamon. So I'm going to start off by motivating this work a little bit by a description of the invasive species problem that we were dealing with in the Salish Sea. Okay, so this is the European green crab. I think maybe some Oregon folks might be familiar with it because it's been a menace on some of the estuaries here as well. It is a voracious predator blamed for the collapse of the soft shell clam industries in New England and Nova Scotia. The various effects it has, it outcompetes local species for food, destroys habitats, and generally has caused, they estimate $22 million worth of damage in the United States every year. And the bad news was that we had the first sighting of European Greenland in inland Washington confirmed in 2016 through a citizen science monitoring effort. So that was in, oh, here's my, do we have the laser pointer? Yeah. So that was in Westcott Bay. But while green crab was new to inland Salish waters, but not to the West Coast. From 1989 to 2000, the green crab spread steadily north. So it was first identified in San Francisco Bay in the late 80s, and then first identified in 93, around 94 in Humboldt Bay. And you just see this like steady progression of the species spreading up the coast, and had filled up, by 2000 had hit Vancouver Island. And while it had hit Vancouver Island in 2000, it did not enter the Salish Sea. These circles show where the green crab was found, and the white circles are the survey sites. So they were monitoring for it all in these waters, and only finding it out on out here. And 2011 Department of Fisheries and Oceans Canada investigation found green crab thoroughly established in Souk Basin. Souk is right in here. But they concluded that they came from an infested mussel shipment and not from natural transport. it was a really interesting project where they had moved they were trying to get the soft shell clams away from the green crab. And when they moved the mussels, they had green crab larvae inside and infected the new bay. So it was very sad. And then by 2016, you were finding them further inside. So, and then after 2016, they were found all around these inland waters. So I was interested in understanding how green crab got into Salish Sea from coastal waters. But as a physical oceanographer, I want to step back and first talk about some of my work on how anything gets into the Salish Sea. Because, yeah, so I'm going to give you guys a little bit of a background on that circulation and the investigations we've been doing there, because that ends up being really important in the story of how green crab get into the Salish Sea. So as a physical oceanographer and someone who studied estuaries a lot, we talk about shelf inflow, which is, so an estuary has river inflow and it's got river outflow, and then shelf inflow is the water from the ocean end that comes back in. It's like if an estuary is a mix of seawater and fresh water, the shelf inflow is the ocean water ingredient. But it comes from somewhere on the shelf. So that's when I say shelf inflow, it means it's referring to these waters as opposed to the continental slope or the deep ocean. It's the waters that are adjacent to the estuary. So we were curious about the effect of wind, because the wind in these areas changes seasonally. And we had these two hypotheses that that the shelf inflow is going to be sourced from the upwind part of the estuary. This is based on some idealized modeling work from the 90s. So basically, when the winds are coming from the north, the inflow should be coming from the north. And when it's coming from the south, the inflow will come from the south, but also deeper and denser water during upwelling. And the Northern California current system is a really interesting place to study this, as so the California current system basically meaning the West Coast. But it's in the northern part being the Pacific Northwest. But the Northern California current system has these reversing winds that can affect the transport between all these places and the connectivity. So so during the summertime, we have our upwelling season where the winds are coming from the north, and then ekman transport pulls a lot of that water offshore and brings up cold, nutrient-rich water. And then during the wintertime, it's the opposite. Things are coming from the south and you have onshore transport. So what's happening at the coast is actually a lot warmer. And that signal is expected to show up in all of these little estuaries that live on the side and affect how they can transport between each other. And then so for this bigger transport question, we used, well, and for the crab question in particular, we were looking at the Salish Sea, which is the biggest estuary in the Northern California current system. The little n is for northern, the big C is our California current system. So the Salish Sea is this little pocket up here. So it's between Washington and the Vancouver Island. I guess I was referring to it throughout, so you might be familiar with it. It's a glacially carved fjord, so there's a bunch of deep, narrow channels. It has the entire watershed of, well, this is a picture of the watershed, but it passes from Western Washington and then a lot of BC. So it has this huge amount of fresh water coming into it from all that rain. The mouth between the Salish Sea and the coastal ocean is the Strait of Juan de Fuca. And there's a canyon here called the Juan de Fuca Canyon that cuts through the shelf. So to look at the connections between the Salish Sea and the coast, we use this hydrodynamic model of the Salish Sea. Let me see this. Yeah, so this is a model called Live Ocean. It was built in ROMS for those of you who are familiar with hydrodynamic models. It was built by my PhD advisor, Parker McCready, primarily, but in conjunction with a lot of other people who help work on a lot of different aspects of it to get it right. And he's been working with a lot of data to validate it through the years. But you can see if you look at the picture on the left is surface salinity and the picture on the right is surface temperature, and with some little arrows showing you where the currents are. But you can kind of watch like where the Columbia River plume is going. I think is an interesting thing for your eye to follow, to sort of like see where the water on the shelf is going. If I can replay that. That went, and this is a map of the wind, a little windrows. So when the wind is coming north, the Columbia River plume goes north. And I wonder if when it goes south, you get the opposite. And you get these, and so if we're interested in the Salish Sea, it's interesting to note that there are the Columbia River plume. Oh, here I keep forgetting because this is right in front of my face, but everyone's looking at that screen. But the Columbia River plume makes it all the way into the Salish Sea during downwelling winds. And so I'm going to refer to this as Columbia River plume intrusions, that it's intruding into here. And that's and you can see what happens. So during normal conditions, all the buoyant river flow heads out from these rivers and sort of makes its way out to sea near the surface. And so the ocean water is only coming in through the bottom. And then when the Columbia River comes in, suddenly you have buoyant water headed in because the wind's blowing it in. And so that creates a little pocket where things can get in through the strait. And this is and this occurs approximately 10% of the time in summer and 45% of the time in winter. This is hypothesized to potentially be a natural pathway for invasive larvae to get in. Is like when this is happening, and so you can see how deep the outflow layer typically is. Most planktonic larvae don't dive much deeper than 10, 20 meters. So normally this is like a one-way street and they can't going outwards, and so they're not going to get in. But yeah, we had this hypothesis that during these intrusions that there's this little secret pathway in through the Columbia River plume that the larva could enter the Salish Sea through. And so we wanted to test this hypothesis. We wanted to test, we can look at like how the Columbia River moves, but we wanted to be able to test inflow for the entire shelf and then for some of the green crab species in particular. And the way we did this was was through particle tracking experiments. So I know that some people may be familiar with these, but suppose you have a flow field that looks like this. I just made this one up. I drew it in PowerPoint somewhat randomly, but suppose that this is your currents in the model grid. Like the model is composed of just these different grid cells, and you give something a starting position. You look at what the velocity is in the ocean at that point, and you say, well, an hour later it's going to be here given that velocity, and now it's in a new place with a new current. Oh, well, then an hour later it's going to be there, and you move it forward like that. And you can see if you start somewhere else, you end somewhere slightly different. So this is the basic tool we used to track where things are going. You could do this in a couple of different ways. The way that I did it was with an offline system. So like I generated these background fields, and then I did a bunch of experiments, just like placing different particles in different places and seeing how they traveled around. And so first we wanted to see this bigger, sort of more generic shelf study. So we released, this is like where I released particles. So there's, 100,000, maybe 200,000 particles here. And then I released them at different depths. So every 15 meters of depth. But the colors are here. It's basically demonstrating bathymetry, but this was the deepest particle released. So offshore, they get to like 2,000, deeper than 2,000 meters. Onshore, I contoured where the 200-meter isobath is. Oh shoot, yeah, okay, you do see it. That's not just me. But I contoured where the 200-meter isobath is. And then I wanted to test a variety of conditions. just for this initial transport study. So I did releases every 2 weeks and I tracked for 60 days. There's some vertical mixing here. And then the stuff that gets that I consider like inflow to the estuary from the shelf, it just is stuff that entered this mouth. And then I determined the source based on the initial position of those inflow particles. Okay, and what we found, this was our first hypothesis was that inflow would come from the upwind direction in general. We found, so what you're looking at, so this is, I sorted, so we had all these different releases that sampled different wind stresses. So these dots each represent the mean wind stress, and then I sorted them into which ones were during upwelling or northerly winds versus which ones were southerly winds. I kind of colored them by that. And if they were real close to zero, I just left them gray. And so then I combined, so in these left pictures, I combined all the ones that were blue, all the ones that were upwelling, and made a heat map of all their initial positions. And I did the same with downwelling favorable winds or southerly winds. I took all of these experiments where the mean north-south wind stress was from the south, and made a heat map of all the initial positions. And these are example releases. We found that like there was a really strong pattern of when the wind is from the north and we're looking at what comes into the Salish Sea, it's all coming from the Vancouver Island shelf. And when the wind is from the south and we're looking at inflow to the Salish Sea, it's all coming from the Washington and all coming from the Washington shelf. So it's all coming from the south when the wind is coming from the south. And these are example releases, so you can see like what one release looked like. And during the upwelling, they swirl around in the Juan de Fuca eddy and they come through the Juan de Fuca Canyon. And in general, when you like plot what the mean north-south position is of the initial position of the particles that made it in the estuary, it's got a strong linear relationship with how strong the north-south wind was. I have another couple of like observations bulleted here that the particles do travel approximately parallel to shore when they're traveling along the shelf. So that you might expect larvae to maybe travel the same way. During the downwelling experiment, we found more originated. So if you can see, you can still kind of see where this contour is here. That is the 200-meter isobath, that you get more particles that originated off the shelf and over the slope during downwelling experiments. And then that the Juan de Fuca Canyon was a big player during both the wind stress experiments or both wind stress schemes. And the Juan de Fuca eddy is a big deal as well. And then we also just did a quick look at depth. Of course, a lot of plankton have a preferred depth, or they can do their own daily vertical migration. But for these initial experiments, they were just passive. And so we looked at both the depth that they were at, the isobath they were over, and then the relative depth, which is the depth divided by the isobath. Because we were expecting that the ones during upwelling would come from deeper because of that upwelling process. But we found that they weren't deeper, so that's why we looked at relative depth, and they were relatively deeper. Here's again a heat map that is so this A to B distance is so I tried to take a transect through the where most of the particles were coming from. So during the upwelling experiment, it's across the Vancouver shelf, and during the downwelling ones, it's through the Washington shelf. And then I also have a transect at the Juan de Fuca Canyon. So this is a profile of like what the seafloor at the Vancouver Island shelf looks like is the solid line, and then the Juan de Fuca Canyon is the dashed line. And then the Washington shelf is a little bit more narrow, but where I happen to take the transect. But then the Juan de Fuca Canyon is the dashed line again. Okay, and this is again, these are heat maps of all the ones that were during upwelling conditions versus all the ones that were downwelling conditions, and then this is a sample release. Yeah, we don't need to spend too much time on this, just yeah, but it wasn't what we were expecting. We were expecting to see that sort of like deep water coming up, and instead we saw a lot of this slope water from the surface during downwelling, and yeah, things coming from all over the shelf. And the relative depth is important, I want to point out, just because so for example, there was an experiment done on the Vancouver Island shelf where the shelf water at the same depth was found to be more hypoxic than so the shelf water on the shelf water was found to be more hypoxic than the slope water at the same depth. So if you're looking at this and saying like, oh, this water here is coming from the same as this water, it's like, well, actually, it's probably less hypoxic out there than it is over the shelf because of how things use the oxygen over the shelf. And this data is taken from Vancouver Island, so it's relevant to the area we're looking at. And then I want to talk about this just quickly, but there was we looked at the density also because we were looking for that deep water signal. This is just looking at the physics. I'm going to go back to green crab specifically, which are going to be in the surface at the end, but there was some evidence of well, so I plotted maybe this is getting a little too physicsy, but I plotted the different initial positions as a function of both salinity and temperature, and found that during the downwelling conditions, things tended to be like warmer, but also less salty. I mean, warmer and saltier for the same water, so that they ended up having about the same density, more or less. There wasn't a strong signal, but whether they were colder and fresher versus warmer and saltier reflects larger California current system water mass sources, where you sort of reverse from getting when the wind is coming from the north and the water is coming from the north, you get water from the subarctic versus getting it from the south. So this is just the only reason I bring this up is because I think that this was an interesting thing that we saw, and it has implications for what the temperature of the ocean water coming into estuaries is, which is potentially biologically relevant. But also that during downwelling, we saw a huge signal that was just like, instead of having these nice bendy lines and slants, we saw one that just sort of like stuck out horizontally. That was just clearly the water's all of the same temperature and the different salinities. And that's the Columbia plume water that comes in during the downwelling. So just to wrap up the sort of pure physics part of this, we had what is the effect of wind on the source of shelf inflow? Hypothesized that inflow would come from the upwind direction, just sort of like when wind is coming from this way. When wind is coming from this way, that the water would come in from this way. And then that it would be deeper and denser under upwelling. So did inflow come from the upwind direction? Yes. Inflow came from the Vancouver Island shelf during upwelling and the Washington shelf during downwelling. Was it deeper? It wasn't deeper, but it was relatively deeper, and you saw more slope water than shelf water during downwelling. And then the last question was it denser? And it wasn't denser, but it was less spicy, which is a word they use in physical oceanography to refer to the combination of like temperature and salinity. So during upwelling, when things are coming from the north, it's less spicy water, which reflects that the shelf is composed more of Pacific subarctic water than Pacific equatorial water. So that's a little bit on how does anything get into the Salish Sea. So now I'm going to spend a couple minutes talking about our initial question, which was how did green crab get into the Salish Sea? And we designed a different particle tracking experiment to simulate the green crab spawns as particles using 4 different sites with known green crab populations. So here we had Coos Bay down in Oregon. We had Willapa Bay just north of the Columbia River. We had Barkley Sound is this sound on Vancouver Island, and then Souk Harbor, because this is where the infected larvae were present. And then these were the observations we were trying to reproduce. And we did 1 release per month during the observed green crab spawning season. And we wanted to see sort of like how much the alignment of those spawns with, especially for the coastal crabs, like, you know, if you're lining up with the Columbia River intrusions, like, is that how you're getting in? Or do they never get in and it's just the ones coming from Souk? But basically, we did all these experiments and we wanted to ask, can a larval spawn from that location reproduce the 2016 green crab sightings that we saw? So we released particles during the spring and summer months because that's when the green crabs spawn. There was data from Barkley Sound and Willapa Bay that kind of agreed that these were kind of different. This was like zeal density, and then this was, these are different methods, but they showed that both in Willapa Bay and Barkley Sound that April and May tended to be spawn months, and then again in late sort of July, August. And we ended up using kind of the same dates for everything, because even though they vary a little bit. And then so we modified the passive tracers to simulate larvae by adding a deal vertical migration. So this is, you know, for some example release. In this one, the particles are showing the circles are showing the final locations. And do I have where this came from? Oh, there should be a star somewhere. I think this was in Willapa that shows where they were released. But yeah, so this, we tracked like the salt and temperature of each particle. This is kind of a mess, but I'm mostly just showing you that we fixed them to migrate in the way that plankton migrate. And then we have a rate. So there's a lot of data on the larval settling rate of green crab, but and it depends on the temperature that they experience, or like it's based on lab studies that use different temperatures, and they'll age faster in warmer water and slower in colder water. But it was a little bit hard to control for, because the temperature ranges that they experienced as they went along were not controlled like in the lab. And, you know, it's like, yeah, I don't know. It became really, so we just gave them a range and said, like, you know, were they found in this part of the Salish Sea between 30 days of age, which is the earliest they've been found to settle, and 75 days of age, which is about the latest that they've been found to settle. So here's an example of a time that they were what we called transport success, which was during the late summer of 2014. It was an August release. And here I've got stars that indicate the different release points that we tested. And then we let them go through the water, doing their vertical migration and being pushed around by the currents. And no matter where you released them from, they ended up crossing into the Salish Sea, which we had defined as just like this line. Like if you got past there, you were in the right zone. But not every experiment looked like this. Most of them looked like this, where you can see like, here's the line that we had drawn. And there were a lot of them that just like went directly off. And like down here at Coos Bay, they all just went straight out of the model and never were seen again. But even like, you know, Barkley Sound is not that far away, and it didn't, they all just went out to the coastal ocean, all the larvae. So I included here like the percent of larvae that crossed the line. So this was a successful transport, had 11% of the ones released from Souk, you know, 4% of the ones released from Barkley, 27% of the ones released from Willapa Bay, and 0.2% of the ones from Coos. And then, you know, for this one, you had zeros across the board, except a couple from Souk, because Souk starts like right next to our cutoff line. And still there was almost none that made it in from Souk. So in general, success was episodic, by which I mean it varied by time and location of release. So these were the different releases. Coos had a different spawning schedule because we looked at the evidence for that, and so they had some spawns during the late winter. And overall, the likelihoods were low. Less than 2% of all released particles were successfully transported into the eastern Salish Sea. I did a little like color key to draw your eye to like the big numbers. So the zeros are all white. The gray hatching is when we didn't have a release. Yeah, so these are the different locations, or these different columns, and these are the different months. So the big one of the biggest ones was August of 2014 in terms of just like the ocean being conducive to bringing the particles inside. And then another big one was in August of 2016. But we had a bunch where there was nothing or very little. Sometimes we're clearly more favorable to success. So is this a video? Yeah. So Columbia River intrusions do seem to be a key mechanic as hypothesized. So if you like look at what the wind was doing during that time, I wonder if I can't fast forward. Sometimes there's a little scroll bar where you can fast forward. But anyway, so let's watch this clock tick up until August. Here's the summer, everything's going to the south because it's the upwelling season. Your pointer has an arrow, but I think this is August. Okay, so here's August. We release them, and then right after that, things start turning north. They're in the water for 60 days, and during that time, you see some strong northern winds. So there's the physics pushing the river Plume and pushing the larvae in as well. And this is, I don't know if I want to talk about this figure too much because it's quite bulky and we don't want to be here forever. But this was a time series of both the north-south wind on the shelf and the east-west. So that's the near surface flow speed. So expect like when you see that Columbia River intrusion, suddenly the flow goes from, taking all of the watershed of Western Washington and British Columbia out to sea. You're suddenly seeing the Columbia River coming in. So there's a east-west reversal when the wind is coming from the south. And then I lined that up with the releases from the different sources. So this is Souk, this is Barkley, this is Willapa, and this is Coos. And so this is flow in the Strait of Juan de Fuca, and the green, you guys can see that, right? Okay, yeah. So the green background is when we expect there to be the Columbia River intruding. And you can really see in this first release, like everything is, so the way these lines work is they're scaled, like the line width scales with how many particles are successfully in there. So everything's sitting at this like gray, sad 0 bar, or and the magenta refers to like when they're mature too. So like here, Souk, you get a lot of particles in initially, but they're not ready to settle yet, and some of them wash back out to sea before they can settle. Yeah, so like in this first one, you see everything's like basically nothing, and then when the flow reversal happens, they all get pushed in, like that line gets really wide and turns pink because they're ready to settle. So there's a lot of mature ripe larvae hanging around. So there was a relationship between the Columbia River intrusions and successful transport of particles, although it varied a little bit by location. Souk was able to have success and like get so here there were no reversals, but you still saw successful transport from Souk. But when you had flow reversals, you tended to have more success. Barkley and Willapa very much mirrored each other in terms of success, which is funny. They're kind of like equidistant, one to the north and one to the south. And they only, so you see basically all the releases that didn't have any flow reversals in them, or flow reversals being the flow in the Strait of Juan de Fuca reversing because the Columbia River intrusions were 0. And so you only saw success when the release overlapped with a Columbia River intrusion. And then Coos successes were mixed and had like some successes that co-occurred with the Columbia River intrusions, and some that were not successful. So Coos was the only place where green crabs spawned in the wintertime, and that has to do with them being further south. But you can see like they had these like February and March releases that were like prime time for the flow reversals to be happening, and none of the crabs ended up making their way up there. So and one of our hypotheses was that these wintertime conditions were going to be most favorable, and that wasn't what we saw necessarily, that the crabs that were able to spawn in winter were not necessarily making it into the Salish Sea, that you kind of had to have a variety of conditions line up. Okay, so we had these different locations that we were kind of trying to do a little bit of a forensic study on which of them was the most likely culprit, where the coastal locations spawn sites more likely, or was Souk, the sort of like inland one, more likely. And you can just point to the like highest concentration of particles that got in, but I want to point out that there's other information that might modify these results. So for example, catch rates in Willapa and Coos were very low in the years leading up to 2014. So you can see like this was so Coos is the blue, no, Coos and Willapa are both blue, and yeah, there's like there were just very few crabs there, and Barkley and Souk had very high populations. So when we were doing this experiment, we were treating them all equally, but we can use this sort of like other information to say like, well, maybe it's less likely that the crabs from Willapa and Coos were responsible because there weren't any in those estuaries at that time. The highest likelihood of success for Coos, we look at this and it's like mostly zeros, but there's a couple in 2014, there's a couple in 2016, and then August of 2016 was a huge transport success year. But it's also August is when the crabs have their smaller spawn. They like barely spawn at all then. So it's like, I don't know, how likely is it that was the responsible time? And in general, British Columbia crabs were more likely than Coos, but the late summer times where they could get into the Salish Sea are, even for those crabs, like their smaller spawning season. So I don't know, there's a lot to go into this, and we found ourselves more like, well, really, they could have come from anywhere. And that was our main takeaway, was that the Eastern Pacific is a lot more connected than we hypothesized, and that our management strategies need to be international if they're going to succeed. Because when I went to the Salish Sea Ecosystems Conference, it used to be the Strait of Juan de Fuca and the Strait of Georgia Combined Waters Conference, and now they're like, well, it's one water, which is how the native peoples of the land treated it, and that's kind of the way we're going back to thinking about it. And I think it's like, there's no sense in dividing it between USA and Canada. So the management strategies need to be international for success. But also that I just want to remind everyone that the weather is extremely important for shelf circulation, and it can be highly consequential for estuarine environments and ecosystems. So definitely need to pay attention to that, especially in our region where there's like these different regimes. And finally, I've talked to a couple of people about this, but I just want to put a shout out because I know that OSU, and in particular the Hatfield Marine Science Center, is a great place for biological work. I just started a new UW postdoc doing marine mammal eDNA tracking. The main questions that I'm looking at, we're calling it a marine mammal megacosm, experimental validation and development of an eDNA fate and transport model. So the three goals of this project are: how do molecular detections vary in space and time given a known point source? Can we model the fate and transport of eDNA molecules for a target species and estimate the time since deposition? And can we hindcast the most probable time and place of a point source given quantitative eDNA detections? And a little bit of background here is that in Puget Sound, there is a population of Atlantic dolphins that's owned by the Navy, kept in a little pen. So we've got this unique opportunity for detecting DNA of species that is not native. So there's no ambient sources of DNA in the water. It's pretty fixed. And they brought me on to help do some of this transport work and try to figure and try to help detect, like, you know, there's not only like the decay of the DNA through the water, but also the oceanographic circulation and how those two things come together to create the final result of how many DNA counts you see in the water. But I've been on this project for about 5 weeks, so I couldn't make my whole talk about this. But if anyone knows about marine mammals and eDNA, I want to chat with you, and I want to get your knowledge and get your email address, and maybe we can talk. Okay, so I've got acknowledgements for the funding of my green crab work, and this was the funding for some of the transport work that I did. But yeah, I'll take questions.
So if we have a question, Elizabeth, you're going to have to repeat it because you on the mic. And I activate the chat.
I actually got one thing in the chat.
Okay.
Okay.
It may very well be active, but no one.
Okay.
Oh yeah.
So if
it looks
you-
over here like the chat is.
If you're online, go ahead and put your questions in the chat.
I can't get my. Maybe I need to change my pointer.
There you go.
Like this over here.
Let me fuss with this. You ask, take any questions.
Oh my gosh. Okay. Any questions from the room?
A couple questions. One, and it seems like, during your time series, that was, kind of like before the beginning of the marine heat wave.
Yes.
And then you had this big span of, releases during the marine heat wave. And how do you think that maybe, transport was affected by that? Oh, first of all, I'm Kim Jacobson.
Hi, Kim Jacobson.
And what we saw is I work on the Newport hydrographic line and Bill Peterson's during the marine heat wave, just, a ton of offshore copepods. So there was a lot of flow, not just south but north, like we normally see. That seasonality was really messed up. And we really just saw this, huge mass of copepods from offshore, come onshore. So I'm just wondering, have you looked at how that might have affected flow and transport on the shelf?
So the question from Kim Jacobson was that these years that I was looking at happened to be the marine heat wave years, that the same years that we believed were sort of like leading up to the green crab, the introduction of green crab larvae in the Salish Sea, were the same years as the heat wave years. And how do I think that the heat wave affected the larvae, because they saw a lot of offshore larvae during that time? Oh, that is a good question. I remember looking at the El Nino index, because that was something that they could, that there have been some studies predicting like climate change into the future. And so we were trying to estimate like how often that like flow reversals and transport that would like sort of like connect the shelf to the Salish Sea a little bit more strongly, or tied to those, were tied to that. And I remember looking at the El Nino index was pretty like it lined up very well. I didn't study, I feel like I've seen so many seminars in the blog, so I feel like I should be able to remember. I think that it is associated with onshore transport, which is the same thing that that drives the Columbia River onshore. So yeah, this is fine. So yeah, when the wind is when the downwelling favorable winds, they push the Columbia River onshore and into the strait. So that is the same, I think, wind pattern that was driving a lot of the that wind pattern was really prominent during the heat wave, I believe. But I wish Melanie Fewings were here because she would really know. I think that was important. I'll say that. Like, I think that was important for both, like, the temperature being maybe favorable and also for the weather being favorable. Yeah.
If you look at your particle tracks and how does anything get to the
Yeah.
Salish Sea, you did have tracks farther offshore than you expected.
This is
Perhaps.
true, although that
Would a different
Yeah.
year have produced different results?
that's a really good question, because so for that, how does anything get into the Salish Sea plot? Those were done, the example year I was using for these was 2017. And that's because it of the years I looked at, it had the most continuous chunks of downwelling and upwelling. A lot of times there's a little bit more back and forth. And so I wanted to try to capture some continuous upwelling events. But that means that this was after the heat wave. So it's unfortunately for that particular investigation, which I think is a really great question. I'm not sure exactly what the
Yeah, so by
consequences are.
2017, we're trying to say, okay, it's getting back to a new normal.
Yeah. Yeah.
So it would be the year before. it would be curious.
Yeah. So I should definitely look at it the year before.
And then the one to get Eddie doesn't really seem to have a big role. I mean, in particle transport or
On the crabs or on everything?
on everything. I mean, do they get caught in the eddy and?
So I don't know how, oh, yeah, so the blue comes out a little light on the screen, but actually, there's a huge amount of recirculation in it. And a lot of and so we were looking at particles that make it in, so eventually they do come out. And you can even see a little bit of recirculation over here. I think, to me, at least during the ones that we tested, it seemed like a lot of the particles did go through that eddy and were sourced from that eddy when they came from the north. So that ended up being like something that a lot of stuff passed through, which is very for hypoxia, for intrusions of hypoxic water that you might be interested in seeing where those come from. The Juan de Fuca eddy is a huge hypoxia hotspot. I think because of the high retention in that area, it doesn't get refreshed very easily.
That only really happens.
Yes, it goes away during the winter.
Is there any questions online?
Yeah, we have one question. What does the genetic evidence suggest as far as Souk or the open coast being the source of the Salish Sea samples?
I think I saw that this was a question. Oh yeah. So the question from the chat was, what does the genetic evidence show about how of the crabs that made it into the Salish Sea and how they were connected to the different populations? I think in particular the Souk samples. I think I saw that there was a question in the chat before Cinnamon took over. I think I saw that this question was from Sylvia Yamada.
Yes.
She was she's really big in the green crab tracking community, and she knows that I know the follow-up work to this by Carolyn Teepolt. So yes, that's an important part of the puzzle that was not yet published when we were originally doing this work, but has been published since, so we can talk about it. So these results, we tried to simulate the crabs to give them the best chance to succeed in terms of like getting out the door. of these estuaries. So a lot of these releases were simulated near the estuary mouths. Souk actually has really high retention, it turns out, and like for the places that the crabs are, they end up being a very isolated population, and we know this because they've been Carolyn Teeple took a bunch of genetic samples and found that that there is extremely high connectivity between a lot of the different infested estuaries, both in the coastal ocean and the Salish Sea, but the Souk population is a bottleneck. And they're very like concentrated, and they're very, I don't know what the genetic term for it is, but they're very inbred. There's not a lot of connectivity between that population and the other ones, that they may have, that they did originate from the Vancouver Island shelf, but now that population is not very connected. So the Souk, so when I was originally telling the story, and I said like they had been on the coast for a while, and then they were moved into Souk anthropogenically, and then shortly after that they were found inside the Salish Sea, it sounds like, oh, so they came from Souk. But there were a couple of reasons to believe that that was not the case, and that they came from the coast, which was what motivated the experiment. And then the genetic work came after that said, you know, they probably didn't come from Souk. So that's, yeah, so that is a really interesting part of the story that I think Carolyn Teeple has followed up with in years since. Although I'm not sure how much of that, yeah, I'm not sure how much that's published, but if you're interested in that, and that ties into my new postdoc work as well, in terms of looking a little bit at genetics. Yeah, that's a really cool part of the story. Okay, yes, in the back.
This is a really dumb question, but I didn't understand particles.
Mhm.
Are these real things that can be lost, and how do you? They're not. They're. How do you know?
What do you mean can be lost?
What's for a loss rate?
Oh, so for the two experiments that I showed here, they were conserved. So we were just tracking, you know, if something started here, where would it end up? All things being, you know, all things being equal. So we were trying to highlight possible transport pathways. These were not intended, and we included the vertical migration because that exposes the crabs to different currents. You know, as I kind of showed with the initial flow reversal plot. Yeah, so it matters whether they're, you know, at 20 meters depth or at 200 meters depth. That said, they are not really meant to, if you follow a single particle, it's not going to be it's not supposed to represent the life of one crab larvae that may be eaten by a salmon or something. So we didn't include mortality or other things that might affect a real crab larvae. We just tried to get the general where currents might take them, and just answer that part of the story. Other parts of the story, like how many were at the source initially, and how much success would there be given, you know, predators in the water for a given year. Those would modify our end result. This is just we focused on this part of the story.
So the second part of that is that, of course, the crab want to be not stupid. They're introduced. They don't know they're not evolved here, but their loss rate would probably be much less than, say, a particle, random particle.
What do you mean their loss rate?
They might actually know how to get around. They definitely find their way back to the estuary, so if they can do that, then there's something that they're detecting to get them to a place that the circulation of the water, of the particle in the water, is not the same as couldn't be the same, isn't likely to be the same as, say, the larvae. So it's not too funny what you're saying. But the question is, if you were looking at your time period and the circulation pattern there, to say the average or the range or something, how much variation is there likely to be?
Yeah, so the question is, crab larvae aren't totally passive, and can control where they go and have an invested interest in finding estuaries, getting into estuaries in a way that passive particles do not represent. So, so one of the things that I would say that they do have swimming speed. Their swimming speed, when we include the daily vertical migration, it's because their swimming speed is comparable or can overcome almost any vertical current in the ocean, because the vertical currents are small. Generally, things are flowing side to side instead of up and down. I think you can see in some of the pictures that they do get occasionally mixed out of their preferred range, but then they can return to the range that they want to be. They could overcome sort of like for turbulence. But when they're swimming side to side, I don't think that a crab larvae can actually overcome ocean currents in terms of like swimming faster than 20 centimeters a second or something like that. So we did choose to neglect the horizontal swimming speed and assume that it was going to be dominated by the ocean currents. The other part of that story is that on a small scale, once they get into the estuaries, they may have more control over where they land. So again, we tried to be broad and conservative in how we both modeled them getting out of the estuaries and how we counted them as successfully reaching the Salish Sea. But you're right, once you get to the scales on which their horizontal that they can kind of steer where they go, or the scales over which that they can swim faster than currents, like we don't model that, or we did not model that. Yeah, did that?
It's like the particles can be at minimum.
Yeah, yes, but
No, because they also experience mortality, right? That plays against them, and horizontal swimming is really in later stages, right? They don't start very strong swimmers.
yeah, this is pre-megalopy. It's yeah.
And I mean, some of what I've read is like that they might follow the sun, right? But that reverses, right? So the horizontal orientation is much more difficult to attain for the larvae than vertical, right? I think Shanks tracked crab megalopy off San Diego, and right, it was like, mostly they attached onto the diver, right? But I think they couldn't really follow the sun, which meant sometimes they were going east something. So is there other crab behavior that's more orientated until they're really close to their environment?
So I'm actually gonna stop our conversation here for just a second and say thank you to our presenter. Thank you very much for coming.
Oh, you're welcome. It was a pleasure.
And then allow the room conversation to continue, while we say thank you very much to you, and we go ahead and end today's presentation. Sounds like there's some follow-up conversations that might be happening. So I'm hoping that helps. Again, thank you, everybody, and thank you very much.
Thank you. Thanks for having me.
I didn't mean to interrupt.
No, I think that's
Long
great.
back.
I saw the time and we can have passionate. Right. I have 4 people are exactly to have this conversation. Why are you?
All right, for everybody online, thank you so much. We're going to end the conversation now. Elizabeth, did you have a contact information on that last slide?
Yeah. Oh, yeah, makes sense.
Yeah, so for anybody online, you're welcome to check out the screen that's up now, and the contact information is there. If you can't see it, contact me and I can make sure I can connect you if you want to do follow-ups. And again, everybody, thanks for joining us.